The virology section allows you to take the data that was produced by the pipeline and visualize it using this interface. This interface shows the reference genome of the virus, the RNA virus, and then it shows all of the passages here. This right here is coverage, and the coverage is represented by a line according to every passage. So you can add and remove passages to display only the ones that you would be interested in seeing. You can also go in and check more specifically the coverage by number. And it is important because our calculation of mutation rate is based on the coverage. You can also change the bin size and see higher resolution by bin size. The bin size is by number of nucleotides. All of the information about every aspect of this visualization can be seen by hovering over the I button. Now, if you want to look deeper, for example, this area or this area, they display that there is a pretty high rate of mutation. And the high rate of mutation is also adjustable by this slider, so you can make it more sensitive for an area of mutation that you want to see. Of course, more mutations are going to be revealed by lowering the red area. Now, if we want to check this area, we are opening now a view that shows the nucleotide level of resolution, and you can also move by bins using these arrows. Now, once we've seen this area of interest, we can hover over it and see which mutations occur, which nucleotide it is, which position, the sequence, variability, coverage, and like I said, which letter the mutation occurs. So now we can close this and also check the different types of mutations. First of all, we are displaying synonymous and non-synonymous at this point, so we can turn one or the other off to check which remain. And now these are the types of mutations that our system calculates based on adaptation. To simplify, beneficial are the areas that are beneficial for viral adaptation to the host. Neutral means that they occur randomly. Detrimental displays the mutations that are not beneficial to the viral adaptation. And lethal means those mutations actually change the amino acid to a stop codon. So those are special types of mutations. Now, if we only look, for example, at the beneficial ones, and we can see how these mutations are mapped on the 3D structure of the protein, in this protein right here, and we can make this image bigger. We can take a look at how these mutations are mapped on the 3D structure, and we can also play with the different views over here. So this right here shows me the amino acids colored in the same way that I see them right here, and I can also hover over them and see them displayed on the 3D structure. And as you saw, I can also color by ratio, by mutation rate. Now I can also render in spheres if I want to look at that view. So for example, if I'm interested in this area, I can see right here, and you also see the area that is covered in this view. And then displaying different types of mutations is done over here. So for example, I see this cluster right here that could be of interest to me, and that is a way to see the data in a more visual format. Now you can also read a little bit about this protein, and by clicking on this you actually see the copy and paste version of the amino acids if you want to take it further and research. So This shows different proteins. The different proteins could be again viewed. You could see subproteins, and we can also see this help that explains a little bit more about this visualization tool.